Hello, everybody out there. It's time for asynchronous recorded lecture 11 for Bio 145 Microbiology. This lecture is scheduled for Monday, February 9th of the year 2026. And today's topic is the first part of infection and disease. All right, let's do a little recap though first. So let's start recapping from where we ended last lecture. We were just finishing up that lecture. We said recombinant DNA technology is very powerful and it's led to different types of things like collagen that's made in yeast that can be put into people's bodies, either for therapeutic or cosmetic purposes, and other really interesting things that do good. However, there are some problems with recombinant DNA technology. Okay, number one, if you have a recombinant microbe and it's sent in the environment, often it doesn't survive very well. It is outcompeted or gets eaten by the natural microbes that are out in the world. And the reason is because when a microbe is taken to the lab and the DNA is changed and a lot of modifications are made to it, it becomes weaker. It doesn't have a lot of the advantages it used to have. So when you stick it back in the wild, it doesn't do very well. So that's kind of like the little cartoon here on the left suggests. And finally, there's a fear in the general public of recombinant genetically modified organisms being used, for instance, especially in foods. All right, and that's kind of exemplified here by this takeoff of a frozen dinner. So although genetically modified organisms can do a lot, they're used for drugs, which, for instance, insulin, which people are perfectly happy to use. When you talk about things like food supply or in the environment, there's a fear there, and that's kind of stopped their use there. All right, well, that ends the topic on biotechnology, commercial use of microbes. And now we're going to start a new topic, and it's a complete change. We're going to focus on infection and disease. And today we're just going to begin a general lecture talking about a lot of terms for infection and disease. And we'll be using these terms for basically the next 18 lectures. So I know it's kind of dry today, but it's important stuff to give you a background. So let's first start with the definition of the first keyword, disease. Disease is just any deviation from the normal healthy state where a person feels sick and they can't carry out their normal functions. That's disease. We're going to focus on infectious disease. I'll get to the word infection in a second. And that usually means microbial based diseases. Now there's other kinds of diseases like congenital diseases are the ones you're born with. Like some people are born with muscular dystrophy. But we're not looking at congenital diseases. We're looking at things that usually microbes come into the body, inhabit, and they're infecting you, and they cause this perturbation, deviation of the normal healthy state. So that's disease. Contamination is a word meaning that microbes are present in something inanimate, meaning non-living. So contamination means the presence, growth of microbes in something that's inanimate, like water or oil in this, as this in this picture up here. Okay, fuel. All right, we'd say this is contaminated oil there. You don't say a person is contaminated. No, the host is contaminated. Infection means, yeah, if you have microbes present growing in or within a host's body, a person's body, you say, oh, that person is infected now. So the body is being inhabited by some type of organism, which can be a parasite, a commensalist, or a mutualist. So infection just means taking up residence. The microbe, in this case, is taking up residence. It's infecting you. And you might think, oh my gosh, this is starting to get too much already. You start to, well, this is only the beginning. Be patient. Stick with it. It'll be worth it. Now, not all infections are bad, all right, because it really depends on the microbe we're talking about and its relationship to your body. So here are three types of symbiotic relationships between a microbe and a host, okay? And think the host is you. So in this case, an organism, that's going to be a human. Let's just say that, human here. And organism one is the microbe, okay? So mutualism is a symbiotic relationship where both benefit. For instance, the bacteria in your colon, they benefit because you're basically providing food, warmth, things like that. And you're benefiting because a lot of these microbes actually produce things like vitamin K for you. So that's a mutualistic relationship of an infection. Commensalism is the microbe benefits, but you, organism number two, are neither hurt nor helped. And that's, for instance, like the members of the genus Staphylococcus that are on your skin. All right. Most experts would say you're not really helped or hurt by them. All right. But they are basically get a lot of things from you. You give them a place to live. You give them temperature. You provide food for them. So that's commensalism. And the third is parasitism. We've already talked about that word, right? All right, parasite. So the microbe benefits definitely, but you are harmed. And that's like some of the really bad infectious disease we're going to talk about, like bacteria that cause tuberculosis in your lungs. They're parasitic. They're pathogenic, okay? Pathogen because, again, they're leading to disease. So just kind of reminding parasitism, pathogenicity, very similar. All right. Now, whether a person is infected or diseased depends on the relationship between the host and the microbe. That's pretty obvious here. And that's why you can be infected, but as in the case of a mutualist, if you're infected, you're not going to be diseased. You're actually being helped by that infection. So you got to think about what the relationship is when you're talking about the microbe with you. So let's kind of look at this next picture. And here, what you have is you have these microbes, and they're already dyed pink. Right? You can see them. Looks like there's some type of bacilli. All right. And those brown cliffs in the background, actually some respiratory tissue. All right. They're respiratory tissue. And you can see that well, you've got the microbes, they're living on the respiratory, your tissue, and you're thinking, oh, they must be bad. Well, again, most of the microbes in your body really and truly are mutualists or commensalists. All right. In fact, we have a term for words for microbes that live in your body, and they're called the normal flora or microbiota, and also known as microbiome. There's quite a few words for them now, so kind of get used to seeing them. I like to use normal flora because that's kind of been the standard one that's been around many years. So microbes that infect your body without usually, okay, and we're going to get in this later, usually causing disease, that's your normal flora, your microbiota, your microbiome. And there's about, on the average person, there's about 40 trillion. So, that's like 40,000 billion microbes in your body, all right, that are part of your normal flora. That's pretty amazing when you think about it. So most of these microbes are commensalists. I don't know how they determine that. You can count 40 trillion of them, but most of them are commensalists, meaning they are benefiting and you're really not hurt nor helped. Now, where are the normal flora? Well, they're at the interface of the environment and you. So think of the environment, meaning air. Well, where's air? Well, there's air against your skin, air against your respiratory tract, and air, for instance, in your digestive tract. All right? Food is part of the environment. Where's their food? In a digestive tract. Okay? So when you have microbes at the interface of the environment and your body, those are usually your normal flora. When do you pick them up? Well, you pick them up pretty much right after birth. When you're born, you're considered sterile, meaning you come out of your mom's body and you don't have any microbes on you. But instantly you start picking them up from the air, from rubbing against your mom's body. You'll pick up microbes from her skin. Your Aunt Bertha kisses you. You kind of get the idea, all right? You start eating things, you pick up microbes. And a lot of these will become your normal flora. Now, a lot of people think, well, do you have normal flora in your blood? And the answer is no, folks, absolutely not. Normal flora should not be in your blood. If there are normal flora, that's probably that's a disease state usually. So I just want to make that clear. Sometimes you hear these crazy, nutty people on the 2 a.m. in the morning and these infomercials talking about helping normal flora grow in your blood. No, that's not true. So the normal flora that you have really depends obviously on your environment, the ones you have on your body. And there's been studies that say that basically if you can differentiate two people, not by looking at them, but by just looking at their normal flora, because everybody has slightly different environment. All right, everybody's, meaning like for instance, the food you eat is part of your environment, and that's going to affect your normal flora in your gut. And everybody's eats slightly different diet. Classic example where they've done studies on normal flora is they'll have twins. So the twins will be born, come out of the body, and they're split. Maybe the parents divorce, and each takes a twin, and the twin live in different areas. Now, they may be genetically identical, they're identical twins, but they go to different areas, they interact with different people, fed different things, and then a couple years later they check, and the normal flora are very different between those twins because they've lived in very different environments. Versus if you have identical twins and they're always together, live in the same home, experience the same things, their normal flora are slightly different. Okay, no one's got the exact same normal flora as the other, but they're pretty much the same because the environment is fairly the same. Okay. Now, normal flora, when you hear about them on TV, things like that, they're usually depicted in a very good way, okay? You want to, they're often referred to as probiotics, okay? All right? These normal flora you buy in capsules, certain foods, oh, they're probiotics, they're good for your body. Yeah, most of the time. But normal flora is kind of like the mob, all right? There's the mob. And the thing is, the mob, you can imagine, they won't hurt you. They might even help you as long as they respect you. But if they see a weakness, they will take advantage of you. So as long as you're tough and strong, they respect you. They don't hurt you. But beware of looking weak. All right. So that's the idea. They're good, helpful, if you're respected. Now, also, the normal flora need to respect you. They need to also be in the right area of your body. And they can't overgrow. They need to be contained in the right area of the body. So think about that. Not the mob now. Think of normal flora. Respectful, in the right area, and contained, i.e. in the right spot, and only in that right spot of your body. So when do they get dangerous? When those are not fulfilled. Let's give me some examples. Lack of respect. If there is a weakness in your body's normal defenses, and we're going to use these general words, immunosuppression or immunocompromised. We can use those interchangeably. Someone is immunosuppressed or immunocompromised. All right? For instance, let me just give you an example. There are normal flora in your respiratory tract. We just saw a little picture of them. As long as you have a normal, healthy immune system, for instance, phagocytes, you got phagocytes, we talked about those. Those are those human immune cells that roam the body and swallow up bad things. And normally they're there, and sometimes they'll swallow up the normal flora there. All right, that's their job. They're just running around, those phagocytes, and they're eating up normal flora, and normal flora aren't causing you problems. But there's examples where certain things make you immunosuppressed or compromised, and the number of those phagocytes goes down. I'll just give you a little cocktail. What they found is like when they've been there with their wife for like 40 years or more, and the wife dies and they become a widower, almost always depression, separation, leads to immunosuppression. And one example was there's a decrease in the number of these phagocytes in their body. And when that happens, they're more likely to come down with pneumonia. So these normal flora bacteria, before were totally benign, once the body was weakened, less phagocytes, they felt that weakness, they attacked, led to pneumonia. We'll go to more examples like this later. All right, in the right area is the next one. So if the normal flora go to the wrong place in the body where they usually aren't, that can lead to disease too. Okay, for instance, gut bacteria, all right, they live in the gut. but sometimes they'll get to the urinary tract, especially in women. All right, incorrect wiping after defecating transfers some of those bacteria that were in the gut from the poop into the urinary tract. And guess what? Urinary tract infection and disease. The normal flora of the gut, totally benign there in the gut, get to the urinary tract, cause a disease problem. Wrong spot. And finally, the idea of being contained. They shouldn't overgrow. If they do overgrow, too many of them, now you have disease. Example, and here's something I need to clarify something. Okay, women, you've heard of say, oh, a woman has a yeast infection. She has itching and burning and blah, blah. Well, there are always yeast in a woman's vagina. They're part of the normal flora. They're always there. And as long as they don't overgrow, as long as they're kept in a reasonable concentration, they don't cause any problems, any disease in the woman. But sometimes, for instance, if there's a hormonal imbalance, and we will talk about this later, the yeast will overgrow. They overgrow in the vagina. There's too many of them. And now they start to cause problems. They start to cause the inflammation and things like that. So they're in the right place, but they've overgrown. So these are 3 examples where normal flora can cause disease in the human body. More later. So you kind of get the point of, well, is a microbe good or bad, or how bad is it? Well, you can't say how bad a microbe is. You have to use terminology for that. So the word is virulence, or how virulent is that microbe? So virulent, it basically refers to the relative ability of a pathogen, a disease-causing parasite, to infect a host and cause disease. Its ability. Now, also sometimes, and in a related way, virulence is also defined as the degree of damage caused by a host, caused to the host by the microbe. Meaning, how bad of a disease is it? You know, if it's deadly, we say, wow, it's very virulent. If it's pretty mild, you say it's, mildly virulent or very low virulence. Okay. Usually it's the second definition when you read about it or people use it in the media. Okay. So let's go like a highly virulent pathogen. We say it's pretty much always going to cause, it infects you and a person gets a disease. And we say, wow, they get that disease of that highly virulent pathogen and it can be really serious and deadly. For instance, some bacteria that cause pneumonia and you get bleeding from the lungs and the person dies, okay? Think of that. Or tuberculosis, okay? TB, we'll talk about TB, tuberculosis. A really, bloody pneumonia, all right? All right, those are some, and you can think of some other ones that are really bad. Necrotizing fasciitis, where the bacteria literally start eating up the tissues and just go through the tissues in a short period of time, kind of eat up the person. Highly virulent. On the other hand, an avirulent microbe is, to a normal person, doesn't cause a disease. All right. For example, there. How about the microbes in yogurt? Okay, think of those guys. All right, lactobacilli, streptococcus, lactococcus. All right, those guys, you think to a normal person, they're not going to cause disease. Versus to a normal person, something that's highly virulent is still going to cause disease. All right. Now, that normal person is always a tough one, okay? Always a tough one. Now we get into like little exceptions. So here's the catch. A microbe may be virulent to an immunocompromised person, i.e. someone who has a weakness in their immune system, but to a normal healthy person, it's completely avirulent. All right? So we kind of got to remember, again, we talked about before, the relationship between the microbe and that specific host. All right? So when you talk about, what if you have a pathogens like that won't mess around with you if you're healthy, normal, but will take advantage of you and will damage you if you are weak, well, we call those pathogens opportunistic pathogens. And that's going to be a very common word in this class because they just cause disease only when the host is somewhat immunocompromised. Their immune system is weakened. Now, what are example of people that are immunocompromised? Well, people that have had like transplants. Often, in order for their body to accept the transplanted organ, you have to weaken, lower the immune system so the immune system doesn't reject it. A lot of cancer patients, just because of the treatments they're on, their immune system is kind of in shambles. You'll learn HIV/AIDS patients, their immune system is really greatly damaged. All right. People that are really old, people that are malnourished, or someone who's already got one really bad disease, and then this opportunistic pathogen comes in, they can take advantage of that person's preoccupation with the first pathogen, and therefore the body can't fight the second pathogen. So these are all examples of immunocompromise. It's a big umbrella term. And you can just see in this example on the right here, all right, where these are just examples, don't memorize, these are just different of microbes that are considered, they cause opportunistic infections. If in your body you're normal, you're healthy, they're not going to cause any problems. But if you're immunocompromised, yes, they can lead to a disease. Again, it's like a parrot. You think, oh, that parrot is not going to hurt anybody. Yeah, but if your defenses aren't up, like this poor little dog, that bird's going to bite you. Okay. Now, a little bit more about diseases and infections. Nosocomial, another word we're going to hear about. These are diseases that are picked up, infections that are picked up and spread in healthcare facilities. So in nosocomial, we're thinking a hospital, a clinic, a convalescent home. All right. Now, why do people get sicker in hospital sometimes? So they go in for a hip transplant and they develop a pneumonia. Okay. You might have heard that, especially in the elderly. Okay. Well, why does that happen? Well, three main factors. And some of these are just logical, but you kind of forget about it. Number one, most people that are going to the hospital or the convalescent or whatever, they're fighting something else. They're immunocompromised already. All right? They're weakened. That's why they have to go to the facility. Number two, you bring in a lot of different people that are sick with a lot of different things. So you're going to bring in and concentrate a lot of microbes, pathogens there. So that's the second reason. And thirdly, you got staff and people that are working there, and even patients that are going to basically transmit these microbes to those people. And sometimes the staff in hospitals, they're not the sharpest tools in the drawer, okay? You might think they are, but sometimes, there are cracks, and even really good hospitals have cracks, and people that are not that skilled or really not that focused make really boneheaded mistakes, unfortunately. So a nosocomial infection can be caused by either a virulent or opportunistic pathogen. Okay, I just want you to think that. All right, a virulent or an opportunistic one. Keep going about nosocomial infections. Actually, there's an interesting stat. 10% of hospital patients, okay, just hospital ones, they pick up a nosocomial infection every year. Classic example is a pneumonia. All right. And pneumonia is going to be some microbial infection of the lungs, okay, lower respiratory tract. Again, what are they caused by? Well, we kind of get into more specifics now. Okay, granted, the pathogen can be opportunistic or truly very virulent. Sometimes you get it from other people's pathogens. So they're called an exogenous nosocomial infection. Exogenous nosocomial infection. Okay, for instance, you go to the hospital, the patient in the bed next to you has some horrible bacteria. They're coughing like crazy. No, the curtains aren't that great. And those bacteria, you end up breathing them in. You came in for a hip replacement or a knee surgery, and you now have a pneumonia thanks to your patient, your next door neighbor. All right, or your next curtain neighbor. That's an exogenous, other people's pathogens. However, what's interesting here is sometimes it's your own normal flora. Like I said, the example where the male widowers, because of loss of their wife, separation, their own normal flora sense that weakened immune system and attack them. So these are known as endogenous nosocomial infections. Yeah, the person went to the hospital, they had a problem, they were immunocompromised, and in the hospital, that's when their own normal flora attacked and cause a disease in them. All right, because of that weakened immune system. All right. Next set of definitions. So someone is sick. Well, they're coughing, they're feeling achy, throwing up, pulse, high pulse, fever. Well, these are manifestations, things that are showing you that they have a disease. One word to describe the manifestations are signs. Signs are objective manifestations that you can measure. You can put a number on. You can see it. For instance, the classic one is fever. Okay? Elevated heartbeat. Viral count in the blood. You can put a number on it. On the other hand, another manifestation, and this is, people use this incorrectly, but we still use it, is symptom. A fever is not literally a symptom. A fever is a sign. A symptom is something that cannot be put into a number. It can't be measured. It's something that's subjective. It's something that the patient feels. For instance, I'm dizzy, I'm fatigued, I'm tired. These are all my symptoms. Can't put a number on tiredness, right? So those are the correct uses of the term. I'm really never in a test going to make you differentiate them. I'll usually just say the symptoms and signs of this disease are boom. But for the purist in heart, know the difference. Signs, measurable. Symptoms, very subjective. So next you're thinking, okay, we got those words. How about the phases of a disease? And there are some common phases to all infectious diseases. All right, whether it be a common cold, tuberculosis, COVID-19, botulism poisoning, things like that. What are they? Rabies. And here's an example where someone has, basically this is chicken pox, okay? Measles. But I know a lot of you haven't had measles, but chicken pox some of you have had. So think of kind of something, it's a respiratory disease with a rash, all right? So the first period of every disease is known as the incubation period. It's the time between the entry of the parasite or pathogen into your body and the appearance of symptoms. So during the incubation period, you feel perfectly fine. You are asymptomatic, lacking symptoms. You don't know anything's wrong with you. You're perfectly fine. But the microbe is in your body. It is reproducing and it's building up to the point that it will give you symptoms and signs soon. So that's the incubation period, which you seem perfectly fine though. Secondly is the prodromal period. This is when your body is starting to fight the microbe and you know it. You start having very non-specific symptoms and signs, meaning you can't tell what the disease is yet. You just know you have these things like, oh, you're achy. Well, it could be anything, right? If you're achy. Oh, I got malaise. I'm tired. I'm just feeling hot. Those are prodromal, non-specific signs and symptoms. You know something's coming. Acme, illness, but acme is what we call the acute phase of disease. That's when you have the symptoms and signs that you could usually diagnose the disease by. For measles, oh, now you got the rash. Oh, well, you must have the measles or you have the chicken pox. That's acme. This is like the worst part. It's like acme is the tip of the mountain, right? It's like the worst part. You really have strong symptoms and signs. And folks, at this point, you can get better or you could die. So let's assume you don't die. Let's assume you get better. Let's always be positive. And then you get into defervescence. That's the next period. So here you're just kind of following this around along here. Here's defervescence, okay? After the rash, after acme, now you're at a period of defervescence or decline. I like to use the word defervescence. And that's when the symptoms and signs start to fade. You start to feel better. Literally, it means like you had this high fever during acme, and now let's say your fever breaks. Okay, that's the classic thing. You're starting to get better. That fever's breaking. All right? And then you go through a period of convalescence where you're returning to normal. In convalescence, you're out of bed. You're feeling pretty good. You're pretty much back to normal, but your body is still recovering. It's still maybe repairing damaged tissue and things like that. convalescence. All right. Now, a lot of people ask me this, well, when are you contagious? And really and truly, folks, that depends on the disease. All right. What disease we're talking about. But in general, for most respiratory illnesses, is the person contagious during their incubation period? In general, yes. Are you contagious during prodromal? Yes. Acme? Yes. Generally, yes. Now, when you get to defervescence, probably. And then convalescence, well, it could be yes in some cases. Sometimes it's no in other cases. So, but definitely from incubation through acme, you can pretty much be sure, yeah, the microbes in your body, it's there, and you're shedding it, and you're contagious between those three phases, for sure. Okay? The thing about a little cocktail party now, a little about COVID-19. What's so bad about COVID-19? One thing is, when they've very clearly shown that you are rather contagious, even during incubation period. And that's why they'll say, well, we have to test you to see if you have the virus in you. And you'll say, well, I'm perfectly fine, blah, blah, blah. Well, you could be in the incubation period, and during that time, you are contagious. You are shedding the virus, although you feel perfectly fine. So in COVID-19, definitely, you are very contagious during the incubation period, even before you feel anything. So now we're going to do a little bit of epidemiology terms. Remember, epidemiology thinks about the source, the cause, and the mode of transmission of diseases, okay? So let's kind of follow that up now. So we talked about source, but let's go to reservoir, which is a little more specific here. A reservoir is a continuing source of disease microorganisms in nature. Okay, it's not a fomite. Okay, later we're gonna focus on the difference between a reservoir and a fomite. But a reservoir is a continuing source, meaning that whatever the source is, the microbes reproduce there for a long time, in the long term. It's not just a few hours, a long time, days, weeks, maybe years. The microbes are not there temporarily by chance. The reservoir provides the right temperature, the nutrients, and right level of water and things like that for the microbes to reproduce long term. That is a reservoir, a continuing source. Now, a reservoir can be a thing that's inanimate. It can be like soil. It can just be milk. There are some microbes that their reservoir is they grow in milk long term, because milk has a lot of nutrients, a lot of water, all those good things, right? Now, a reservoir could also be an animal, like a non-human being, or it could be a human being, a person. Okay, long-term place of long-term growth, reproduction for a microbe, a pathogen. Example, we're going to talk about tetanus. All right, you might have heard, oh, you got a cut, you got some dirt, wash out that cut. Why? Because you get tetanus. Well, Clostridium tetani are a bacterium that causes the disease tetanus. The cells and the spores actually reproduce in the soil. Interesting, here's and I don't want you to memorize. This is just examples to help you illustrate the idea. And I'm not going to ask you what Clostridium tetani are at this point. Another type is there were these old flooded tires here. Well, there was a place in Wisconsin, I believed, and there was this dumping area where people would just dump their old tires. It was at the bottom of the hill, and on the top of the hill was this dairy, and dairy had the cows had poop. They had a lot of parasites growing in the poop. There was a rainstorm. The water from the rain brought the poop and all those parasites, and it filled up those tires. And for a long time, those tires were filled with water and poop, and those tires with the water and the poop were a reservoir for those parasites. Okay, just an example. Animals can pass it on to people. An animal can be a reservoir for a pathogen that can even go on a human being. So if you get a microbe, a pathogen from an animal, and it causes disease in you, you'd say, oh, I got this from this animal, and it's a zoonotic disease. A zoonotic disease. One example is why do they tightly regulate what kind of parakeets and other songbirds are sold in the U.S.? Because there's a lot of examples of birds transmitting pneumonia-causing bacteria to human beings. So these So certain pet shop owners would smuggle in these birds that wouldn't get inspected, and these birds would be sick. They'd have these be infected with bacteria that cause pneumonia, and guess what? Person buys the bird, the person comes down with pneumonia. Okay, only buy birds from reputable salespeople. All right. An interesting little cocktail conversation is some people get diseases from armadillos. So there's your armadillo. They look like they have a coat of iron on them. They live in usually desert in areas, dry areas. And anyone want to guess? Well, if you might have heard this, their armadillos are reservoirs for leprosy. Leprosy, all right? And that's a disease where you literally lose fingers and toes and things like that. Really bad. And you're like, well, how did these people get them from the armadillo? Were they armadillos' pets? And actually, there are examples where people got them from armadillos because they were eating the armadillos, okay? Especially in certain parts of Texas. People were rather poor, they needed to save money, and they would capture armadillos and make meals from them. And of course, they'd capture the that was alive. They would contact the armadillo, have to gut it on their skin, and they'd pick up the bacteria and they'd come down with leprosy. So, armadillos, just stay away from them and definitely don't eat them. So people are reservoirs for many diseases. It's in some case that's the only place that the microbe will grow. We've already talked about a lot of viruses. They grow only inside the host cell, and some viruses only grow in human host cells. So viruses that cause cold, HIV, they only reproduce in people. Okay, so people are the reservoirs. A carrier is a very special type of reservoir, and I want to go over this because we're gonna talk about a lot of diseases later where there's a carrier. All right, a carrier for our definition, okay, is a person that sheds the disease agent, viruses, bacteria, without showing any symptoms or signs at that time. Usually a carrier is someone who got infected, had the disease, symptoms and signs, and got over it. But the microbe still was in their body. They were still, even though they felt fine and they were recovered, they were still, what's the word, if the microbe still, they were still infected, okay, by the microbe. And literally, for some people who are carriers, they can have that microbe in their body reproducing for the rest of their life. For some people, they might be carriers for several months or a year, but for some diseases, their whole lifetime. Now, if you look at the book we're using for the class, they define it slightly differently, but this is the definition I want you folks to go with, okay? A classic story is in England. I think this was the late 1800s. There was a group of guys and they were laying pipe. They were putting pipes down for this town. That's a blue pipe being put down. And for some reason, this one guy on the pipe crew kept urinating where they put the pipes. And then the pipes would carry drinking water for the town. I don't know what they were thinking, allowing this guy to pee there. Well, unfortunately, this guy was a carrier for cholera bacteria, and it was in his urine. So he thought he was perfectly healthy, whatever, and he kept urinating, and his bacteria were in the urine. They get in the drinking water supplies. Guess what? People came down with cholera because of this carrier. But, if you met the guy, you would never think he seemed perfectly healthy, fine. But he was infected. So let's looking at transmission, and these are again slightly different from the book because I just kind of like these pictures and they fit better. And if you look at a lot of other books, they follow these categorizations. So I'm going to go with the categorizations, the more what I call the conventional categorizations. So number one, if you transmit a disease, okay, we're still in epidemiology, you can think of direct transmission. It means going from the old host to the new host. When you say direct transmission, you want to think of close personal contact is involved. The direct exposure from one's body fluids to the other. So people are kissing, having sex. Mother's milk, for instance. The new and the old host are in the same space at the same time. Okay. Direct. In droplet transmission, we're saying the microbes are going to be in these airborne mucus droplets, all right, and they get inhaled into the new host. Now, you don't have to be direct because the new host and the old host don't have to be in the same room. For instance, the host, the old host, the one who is infected, can sneeze into the air, all these respiratory droplets, leaves the room, those things are just floating in the air. And then this new person comes in, they're uninfected, they come in and they go, oh, breathe that good air. And they breathe in those respiratory droplets. So that's a droplet transmission. All right. So that's a droplet. That's a second way. So we've seen direct methods up here. All right. And droplet, which isn't in this picture. And then we're going to go to the next one, which is indirect. So are you afraid? Are you afraid of these? These are indirect. Looky here, a doorknob, towel, a syringe, someone serving food, someone who works for dun, 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 dun. How'd that get there? Well, these are all involved in indirect. So here's indirect. We're looking at them. All right. There's an intermediate carries the agent from the old host to the new host. All right. Some intermediate. So it can be food, water, soil. Those have they basically went from the old host. Let's say they were making a sandwich for you with their loving hands, or tossing that salad with their naked hands, and they've got the agent on their hands. It goes into the salad. You eat the salad. Guess what? You get infected. Same thing with water, soil. Now you can also have with contact with a fomite. Fomite's an an inanimate object that carries the host disease, that carries the disease organisms, but only for a short term. Okay, so that can be like a doorknob. All right, someone sneezes, someone's sick, they sneeze on the doorknob. The doorknob has the viruses on it. You then go and, but a little while longer, a little later, touch that doorknob, rub your nose. Now the viruses have gone from your hands to your nose. The fomite was the doorknob. All right. Here in our little picture, contaminated objects, right? Inanimate objects where you're going to find these microbial agents. Okay. Another example, classic one is what's the number one fomite in a hotel room? A lot of people say phone. No, it's not phone, because most people have their cell phones. It's actually the remote control for their TV, because a lot of people touch that remote control of the TV, and housing staff usually don't clean that. So that's a pretty bad fomite. Yeah, doorknob would be bad too. Toilet, yes, of course. How about an airport? What if someone touch a lot and then it's not cleaned very much? It's actually those ticket kiosks, those where you push the screen, you touch the screen. Those are really, really bad in terms of having a lot of different microbes on them, pathogens. Yeah, they've done those studies. So kind of stay away from those, or try to sanitize them before you use them. So we're going to talk about, we're going to do a skit in the synchronous that'll kind of also exemplify some examples of fomites. Now, why are they short term? Though we say it carries only short terms. Remember, reservoir is long term. Why are fomites short term? Why do the microbes only last on them for a short while? Why is that? Because fomites think doorknob, towel, TV, remote control thing. There's no nutrient, right? There's not much water. It's not the right temperature. So the microbes can't reproduce there long term. They're only there a short time. So that's why it's a fomite and not a reservoir. Okay. Next one is vectors. Vectors can also transmit things. This is another example of a kind of an indirect transmission. So animals, especially arthropods, can be vector, meaning organisms that spread agents between major hosts. So and usually these things are kind of like a bug that'll transfer something from a horse to a human, or some type of bug that'll go from a bird to a human. So the bird and the human are what we call the major hosts, and we saw that vector is that bug. All right, humans are not vectors. Humans are not vectors. So when we talk about vectors, we're thinking again arthropods. Okay, insects, spiders, stuff like that. If it's a mechanical vector, the agent is externalized. It's actually on the surface of the vector, the bug. For instance, a fly carries pathogens on its feet. All right, so the fly lands on some poop, and then it picks up microbes on the poop, and then it lands on your tongue. Okay, and all right, or it goes into your nose. It's a mechanical vector. It's on the outside of its body. It's carried the microbes on the outside of their body. On the other hand, if it's a biological vector, then that arthropod, that insect, that bug, is literally infected. The microbe is inside its body fluids. And in order for it to give you it, it's usually got to bite you, okay? Because it's inside its body. So the vector has to bite you to transfer it. For instance, mosquitoes carry pathogens in their saliva and they transfer by biting you. My trick to differentiate is if you can take the vector, that insect, that spider, whatever, and you put it under a little shower and you wash it, and if you can remove the microbe by washing it, then you know it's a mechanical vector. If you can't wash it off, meaning it's inside its body, then it's a biological vector. So keep those differentiated in your mind. This cartoon just exemplifies this. Here, a mechanical vector, basically, the fly picks up, let's say, bacteria from the poop, transfers it now to the food, person eats it. The fly was a mechanical vector. Here, we're saying that we have a person, she gets microbes from a biological vector, okay? It spreads through her blood. And then the second mosquito bites that person, and now that mosquito picks up the microbes from her blood, and it also becomes a vector, and now it can pass it to another person. Okay. How about we do a quick question to kind of test it. You and your roommate Romelda share a towel in your bathroom. She has a fungal rash on her face. Now she's contracted because the towel is a what? So thinking about this, you can pause me right now, but I'll tell you the answer. Okay, towel is not living. All right, it's a dead thing, so it's not a vector because a vector's got to be some kind of bug or insect. So it's either a fomite or a reservoir. Well, it's a towel. Towel is inanimate. Towel doesn't have food and all the right things, so it's probably some type of fomite. Okay, it's not a reservoir. It's a fomite, so it's got to be B or D now. Okay, it's not a vector, not a reservoir. And then you'd say, well, it's indirect. That's an indirect thing. The fomite basically carried it from Romelda, then to the towel, then to you. So it's indirect. So this is correct. Okay. Why don't we take a break right now, and we'll come back for part 2 after our break. Thank you. Alrighty, back from your little break here and mine. Let's talk about some more epidemiology terms that have to do with the spreading of a disease. So first of all, you might have heard the word recently a lot in the news, endemic. They're saying COVID-19 will become endemic. And that means it's a disease that appears at a very steady, low level constantly in a certain place. Okay? There's no surge in the numbers anymore. It just constantly, maybe 300,000 people a month get it throughout the U.S. Okay, it's endemic. Endemic is also an example of malaria in the Philippines. There's a slow, low, kind of steady level of people getting contracting and getting infected with it. That's endemic. If it's an epidemic, well, that's kind of what COVID-19 is right now. And it means there's been a sudden increase in the occurrence of the disease above a certain expected level. So there's a surge. So there's a flu epidemic in the U.S. now. About this time of year, around February, for instance, there's usually a lot more flu around than other parts of the year. So we say there's a flu epidemic in winter. A pandemic means an epidemic that's worldwide, and COVID-19 is an example of a pandemic. This huge increase in the occurrence of the disease at a worldwide level. And finally, on the other scale, if you want something like a localized epidemic, That's as an outbreak. It's like a condemned epidemic, just like in a city. So here was a picture of people in this small city where, let's say, there's been some disease that's localized and people are taking unnecessary precautions. Okay. We're not done with terms yet. Incidence is the next one. This is an important one because you hear this in the notes a lot. Incidence is basically a number of new cases of a disease in a certain area, okay, usually per a certain number of people during a time period, like during a year. So it's like the number of people who have come down with that disease in a certain area over a certain period of time. These are new cases. So they'll say, in this area, the incidence was 20 out of 100,000. So last year, out of 100,000 people, 20 came down with this disease during that year, between January 1st and December 31st. That's an incidence, new. Prevalence, on the other hand, is the total number of cases of a disease, new, and here's the big one, already existing in, again, a certain area over a certain time period. For instance, we say out of 100,000 people, the prevalence is 250. Now, what's the difference? Well, this is prevalence is the number of new cases during that year, along with the cases that existed before, meaning before that year. So you'd say, okay, in a given year, the incidence was 20, the prevalence was 250. That means 230 basically got the disease before this year, maybe 1 year or 2 years or 3 or 4 years before. Okay, and it doesn't include the cured. We're just working about people that are actually sick at that time with the disease. All right. So incidence of AIDS in 2000, 37,000 came down with it between January 1st and December 31st, 2006. The prevalence, though, was 1.1 million, meaning, about 1,060,000 people that had it in 2006. They came down with it before 2006. That over a million caught it in 2005, 2004, 2003. But they still had it in 2006. Thus the prevalence, the pre to that year. So what does it say about the disease? If there's a big difference in incidence and prevalence, what does that tell you about the disease? If the prevalence is so much bigger, let's look at the next graph. So again, HIV prevalence and incidence. So prevalence is in red, incidence is in blue. All right. So every year, let's say maybe 50 people got 50,000, maybe 100,000 got 80,000. So this is per year. But look, the prevalence just keeps going up and up and up. It's like they're accumulating, right? All right. What does that mean? Well, that tells you this disease is not what? It's not easily cured, right, folks? There you go. It's not easily cured. Basically, you get it and you don't get over it within that year. You keep it around for many years. So you add to the number of the people who are considered in that prevalence category. All right. So you've got it in 1982, but in 1990, you still have it. So basically, you add to the prevalence of it in 1990 because you weren't cured of it when you got it in 1982. So that's what incidence and prevalence can tell you a lot. If the incidence equals the prevalence, what does that tell you? It tells you that basically if you get it, you get over it pretty quick, right? So within that year, if you got it, you got over it. You didn't carry on to the next year. Well, that's one thing. The other thing is you got it that year, you died that year. So it's a disease that kills you really fast. So by the time next year rolls around, you're not around to count. So if the incidence equals the prevalence, that means it's a disease that's either easily cured or kills you. A little more definition. Primary infection, okay? If it's a primary infection, that means you were healthy and suddenly you got sick. This is my primary infection. A secondary infection is basically something that afflicts a person that already has a primary infection. So they're weakened. So a secondary infection is often caused by something that only makes you sick if you're weakened. So it's often, these are what? Opportunistic. Very good. Opportunistic infections. All right. Secondary infections. Morbidity, big name here. So this is the total number of ill people in an area divided by the total number of people at a specific time. So it's like morbidity is, how many people of that certain number actually are sick with that disease. It's similar to prevalence, very much is morbidity. Mortality, be very careful here. Mortality is the number of people who die from that disease divided by people who have the disease. So it's not the total number down here. In the denominator here, it's the number of ill people. So it's of the people who have the disease, how many die? That's mortality. Morbidity is of the total number of people, how many are sick. Okay, so keep that separate in your mind. For mortality, let's just talking about, you're hearing a lot about COVID-19, how serious it is. So the mortality rate of the flu is about 0.1%, meaning 1 out of every 1,000 people who come down with the flu will die of it. On the other hand, for COVID-19, there's a little argument with numbers, but the mortality rate is about 2 to 3%, meaning out of 1,000 people, 20 or 30 of those people will die. Okay, so you'd say, oh, COVID-19 has a higher mortality rate. Then we get to the R number, and this is bantered about a lot, but it kind of is a measure of how contagious a disease is. But as you're going to see, it's not just a reflection of the inherent properties of the microbe. It also has to do with the host, the people. Okay, R number depends on a lot of things. So R number, R is that official word, it's the effective reproduction number. What does that mean? That's just the average number of people that a one infected person or reservoir will pass the disease agent to. So if I'm sick and I pass that agent to 2 people, the R number is 2. On the other hand, if I'm sick and I pass it to 10 people, the R number is 10. Okay, you get it. So you get the idea kind of how contagious something is. Now, why is it important? We're here to think about it. If the R is less than 1, that means each existing infection causes less than 1 new infection. All right? So that means the disease is going to die out after a while, because the one person who is sick is not replaced by one sick person. It's replaced by half a sick person or 75% of a sick person. So, secondly, if the R equals 1, that means each infected person leads to 1 new infected person. So it's very stable. The disease is going to stick around and get stable. There's not going to be an outbreak or an epidemic, but it's going to be kind of endemic, right? That's kind of the idea. On the other hand, if the R is more than 1, that means one person will lead to infections in more than one person. So one person gives it to 2 or 3. Now, that means this has the possibility of becoming an epidemic or an outbreak, right? A huge surge. Let me just give you an example, okay? Let's say the R is 1.5 to begin with. So the R is 1.5. Let's go over here. R is 1.5. And let's say I'm going to start with 100 people that are infected. That means each person gives it to 1.5 people. So that means these 100 people will give it to 150. These 150 will give it to 1.5 people. So that's 225 now get it from them. These people, so 1.5 of that's going to be about 350. And you get the idea, right? 100 people give it to 150. The 150 give it to 225. 225 gives it to 350. And that's going to give it to about 500. So you can see this could be an epidemic. On the other hand, if the R is just 0.5, means one person gives it to a half. That means you have 100 people. They're going to give it to 50 people. These 50 are going to give it to 25. 25 are going to give it to 12. You get the idea. And after a while, the disease should die off, and there's not going to be anybody have that disease anymore. Okay, so R is tells you a lot, right? It tells you a lot. Now, R is not constant. It can be affected by a lot of different factors. What causes R? Well, R is basically has to do with the inherent traits of the virus. Let's say the agent is a virus here, like COVID-19. How tough is it? How efficient is it at attaching and entering a host cell? I mean, it's a virus. Some viruses are really good at attaching and entering. Some aren't. So if the R is pretty big, that would at least suggest possibly that the virus is good at sticking around, lasting a long time, attaching to a host and getting into a person's body cell. But here's some things that aren't just of the virus. R number reflects how the population behaves. If the population is wearing masks, for in the case of COVID-19, is not having big crowds gather, there's less of a chance of one person spreading it to one or two or three people. So that's going to bring down the R number. If there's a lot of people who've had the disease and now have immunity to it, or are vaccinated against it, they've got a vaccine, that gives them protection. That's going to bring the R number down also. So that brings it down, that brings it down. However, the R number has a lot to do with location. If population density increases, if people go from living separated out, maybe during the summer everyone's going outside, and then a brutal winter comes and everybody goes indoors and everybody's in the same buildings, they're close to each other, they're more likely going to pass it to each other. So that's going to make R go up. So pretty much, like, you can say, yeah, R has something to do with the traits of the agent, like a virus, but it also has to do with the behaviors of the hosts. Okay, so that's why an R number can fluctuate with time. For instance, this is from the World Economic Forum report. It just said that when Germany, with the COVID-19 pandemic, they instituted a major lockdown, and they found that basically the R went from about 2, what they originally thought at the beginning was about 2, to about 0.7. So there's wow, if people kept that up, that would basically, it would die out. But it increased back to 0.9 after a few months after April because people's behavior laxed. Okay. All right. So we've gone over R. Now let's do R naught, or little r with a little 0 at the bottom of it. R naught. R naught is the reproduction number, but it's for a population that's facing a novel microbial agent for the first time, when they have no behavior modification. So this is early on. So imagine here's a situation where you're really going to mainly look at the qualities of the virus. People aren't going to be doing masking. People aren't going to be having any immunity to it or anything like that. So it's a much cleaner and easier number to get your head around. And it's often used more than R. So it applies when everyone in the population is completely vulnerable to the disease. Like I said, early on when that new microbial agent enters a population, such that no one's been vaccinated, no one's had the disease before, so no one has previous immunity to it, and people aren't doing anything to stop it, like there's no masking or social distancing. So it's a cleaner number, kind of easier to understand. It takes out the effect of what the people in the population are doing. So you see it used probably more than R. Let's look at the next slide to kind of compare the R naughts of different diseases. So here let's compare the R naughts for some various diseases, right? On the far left, we have things that are less contagious. For instance, Ebola, R naught's about 2, so one person give it to 2 people. Then we have the original COVID-19 strain, which we're going back to late 2019, early 2020, and that's about, R naught was about 3, which is okay, but not that bad. Then you can see HIV, SARS, and then here's the Delta strain. So you look in the middle, there's the Delta strain, and that was the one from about last year, and it was about 7, so it became more contagious. Then we have chickenpox, and it's kind of more of a childhood disease, And if no one's vaccinated, no one's doing anything special to stop it, go to 10. Then you have mumps, and then finally on the far right, there you got measles, and that's why you hear about stories like, in history, there's like civilizations being wiped out by measles. It's really, really bad. See how contagious it is. Now, how about the newest strains of COVID-19, the like XB 1.5, the one that's going around right now, the Kraken? Well, I'll tell you that when we're in live lecture. That might be interesting too. Okay, to talk about.
And our last point today is talking about diseases in terms of how long they last uh, and their severity. So we're going to go over the idea of acute diseases versus chronic diseases. You can see the definition there. All right, and like chronic would suggest, chronic just sticks around and lingers. And some chronic diseases you may have heard about is Lyme disease. So you kind of these common like urinary tract diseases that certain people get and they just can't shake them. Epstein-Barr virus. We're going to get more into about this in the next lecture. All right, give you something to think about before then. All right, what makes a disease chronic versus acute? All right, thanks for listening. Take care and stay healthy. Arrivederci.